Hi everyone, welcome back to the channel. This is Dr. Hasik, and today's topic is macroevolution. This is chapter 12 in the Orange Book. So let's get started. So this is directly related to previous chapter, which was microevolution, which basically just a species level, and one of the conclusions we had in previous chapter was the closest evolutionary relationship means most common ancestor. The species under same genus, they are actually more closely related. And then we cover divergent and convergent evolution and evolutionary mechanisms and some evidences. So today we are going to move to chapter 12, macroevolution. Okay, so let's remember some of the student learning outcome in this chapter, which is basically, so you need to also remember all the micro evolution processes you learned in the previous chapter. And remember genetic variation, also important here. And also natural selection as a mechanism, one of the mechanisms like genetic drift, gene flow, or also mutations. And then today we are going to talk about more fossil records and other areas. So basically, a lot of macroevolution. Macro here means large scale evolutionary patterns. So we are not talking about 1,000 years, but we're talking about billions of years, like 3.8 billions of years. So we compare micro, like the small scale versus macro. In micro, it was species level. Macro is about species, and also covers everything with the geological times, fossil records, and comparison between living organisms, and then it might occur due to extended microevolution. Okay, so we need to look at the definition of species in order to understand macroevolution. So species in biology means kind or appearance. Biological species really means defined as a group of populations whose members have potential to interbreed with one another in nature and produce fertile offsprings, and of course offsprings also then can reproduce. So there are we are going to see a number of barriers that separate species from other species, and so very important. Biological species concept based on reproductive compatibility, not physical similarity. Okay, so there are a number of barriers, so that we can actually divide them in groups. So some of them are reproductive barriers. You can see here individuals of different species. They can have actually prezygotic or postzygotic barriers that prevents them, closely related species, from interbreeding. So prezygotic, that means that prevent mating or fertilization between species. Postzygotic, it occurs if interspecies mating occurs, but hybrid zygotes form. Then we see maybe reduced hybrid fertility like you see in this picture, or hybrid breakdown like we see in these rice plants. So pre or post, and also there are barriers like they are it could be like still prezygotic, like habitat, like you can see here, habitat isolation. So land snakes versus water snakes, or temporal isolation. So the mating times of the year is different, or mechanical isolations, like you see here, that basically like snails, they coil on left side or coil right side. So they're mechanical isolation, or behavioral isolations. So maybe like you see here, or in other cases, different species use different sounds, and so not to mate if that sounds, they don't know that specific sound. And And then post-zygotic, they could have sterile situations like you see here in this mule. Horse plus donkey can reproduce mule, but mule is sterile. Okay, so some of the mechanisms of macroevolution. There are a couple of mechanisms that cover here. One is called allopathic, the other one called sympatric. Allopathic means like geological processes can isolate populations. As you see here in this picture here, these two species, group of species of animals, they are separated geographically because there is Grand Canyon separates them. One group left on the left side, the other group left in the right side. Okay. So everything we learn in microevolution can apply here, like genetic drift and founder effect, which if the species left and move to the other side, can actually, since they are isolated there, they can start a founder effect there. So this is allopathic, which is basically geographic isolation. The other one is sympatric. In sympatric, the barriers are not geographic, but they are more reproductive barriers. Polyploidy is one example that we see a lot in plants, especially. Species may originate from an ancient accident during the cell division that resulted in extra sets of chromosomes. Poly means many, ploidy means so sets of chromosomes. Instead of two sets of chromosomes, we see in default like a diploid situations. In this case, there are tetraploid, hexaploid, and extra sets of chromosomes. Okay, so examples, we can see polyploidy examples like in strawberries, which basically in plants we see a lot of examples. Strawberries is one good one. Wheat, like bread wheat, they also have polyploid situations, or some type of watermelons, they have also that too. So speciation, which we know from the Darwin's experiments, his observations and his book, which basically he visited one really interesting place called Galapagos Islands, and in Galapagos Islands he actually observed a number of species that he came up with his hypothesis that biogeography is more important than environmental similarity. So what does that mean is in Galapagos Islands, he's seen these finch birds that with all these different types of beaks compared to maybe nearby islands or nearby mainland. So they specialize and their beak size and changed. You can see here, this is an insect eater one. You can see this guy. This is a tool using insect eater, and this is a cactus seed eater. So they that's actually you can see in this island had 14 species of closely related finches that he observed. So there are other examples comparing to this situation, which basically again we can see actually a divergent evolution. Basically, these birds do have actually a most recent common ancestor, but they actually change. They don't look like each other anymore, actually, like finches of Galapagos. Okay, so they don't look like each other anymore, but their finches are still homologous structures. That means they actually determines like evolutionary. If we do evolutionary tree, they are still in the close branches. Okay. So we can go a larger scale, level, since this is macroevolution. Basically, large scale, we have fossil records that actually that shows strata of sedimentary rocks at, like in this case, Grand Canyon. You can see here that we can actually study which organisms lived in the past from their fossil records. Okay. and then estimate the geological time scales. There is a technique, so like called radiometric dating. So it's based on basically decay of radioactive isotopes to determine the age of this specimen. So carbon 14 is one of these most well-known developed techniques in 1930s at University of Chicago. Now carbon 14 is a radioactive isotope of carbon. It's rare, but it has actually half-life of 57-30 years. So once we know this, and we know that every organisms actually have carbon 14, and they carbon and after the carbon actually left in their bodies, and we can actually measure it with carbon 14 and then determine the estimated age based on this half-life of carbon 14, which is 5,730 years. So 5730. Okay. Again, we can study plate tectonics via geography. You can see all these interactions. So human moved, and they actually migrated. When they migrated plants, animals, other species with them all over the world. So again, biogeography is studies continental mergers, separations, explaining patterns, and studying past and present distribution of organisms. There used to be much more number of species than current 1.8 million species. That means maybe there was 3-4 times more species, so some of them are already extinct. So we can only know about them from their fossil records, and then we can complete all these gaps in our phylogenetic trees. That brings us to this. I go back here. Earth life history. So the current estimate is the Earth is 4.5 billion years Earth formation, and starting with this Hadean eon, 4.5 billion years, and currently we are in Phanerozoic eon since last 540 million years to today, and also we are in Cenozoic era last 66 million years. That's since actually the dinosaurs disappeared, and we are in Quaternary period, and we are in Holocene epoch today. Okay. And also there is in this chapter there is some information about systematics. Systematics is a combination of taxonomy plus phylogenetics. Phylogenetics or phylogeny is basically evolutionary relationship between organisms. So systematics started with Carl Linnaeus in 1780s and his 2 books and more than 5,000 species he actually he classified and named in his books. So today we use phylogenetic trees using homologous features to actually build phylogenetic trees and to try to understand evolutionary relationship between the different taxa. Okay, so homologous characters are the characters that basically there are structures that they may not look like each other. They actually they come from divergent evolution, which basically they used to be coming from they have common ancestors, but they diverse, and then they actually the similarity inherited from a common ancestor, but they don't necessarily look like each other anymore, like finches of Galapagos. Okay, so they are distinct species now, but they have homologous structures. This is very important in building phylogenetic trees. Okay, so if homology reflects common ancestry, we can also use DNA sequences to help actually this because every species have a distinct DNA sequence. Okay, so DNA sequences can be used in these trees. There is another evolution called convergent evolution. In this case, um, uh, so there are similar structures, but that similarity is due to convergence or analogy. They are analogous. It's because maybe because there is a similar environment and they try to actually adapt to the similar environment, and now these two species look like each other, but they don't they don't necessarily have a most recent common ancestor. Okay, so please understand the difference between that. This is convergent, but divergent is the one that we can use mostly in this cladographic cladistics, these trees that we build. So we can build these trees like this here, and is any two branches like these top bottom two here, they will be sister taxa. Any of these green dots here are like common ancestors. This first one here, first line here, like lizards and snakes, they are basal taxa. They come straight out from the common ancestor, main common ancestor. Again, today these are used extensively in biology, building phylogenetic trees in any organisms, including bacteria. So this is there is a little summary here. This is basically microevolution. So origin of species, what are the species we covered? Actually, some barriers we talked about. Just to recap, mechanism of macroevolution. Macro is this chapter here, which is large effects from small genetic changes and classifying organisms using systematics, phylogeny, and cladograms. So this is it for today. Thanks for watching, and please feel free to comment and subscribe. And I will see you in the next video. Happy studying.